We are conducting a collaborative project involving myself at the University of Texas and my staff, and we're collaborating with University of Texas Medical Branch in Galveston, Texas, here in Morogoro with the Sokoine University of Agriculture, and we're also working with a biopharmaceutical company in Morocco who is going to be manufacturing the vaccine that we're going to be using in this project. So when you ask about the project, we are evaluating a vaccine for a disease called Rift Valley fever. This is a disease that affects both livestock and humans, and particularly in the eastern region of Africa, but it's also has spread as far north as Egypt, west to Senegal, and in year 2000 it caused a major outbreak in the Arabian Peninsula. This was in Yemen and Saudi Arabia, and it also has spread to Madagascar. So it's a very important disease, and one of the approaches to preventing the disease is a vaccine. At least we think a vaccine is going to work. So we have a novel vaccine that was originally manufactured or produced in the United States, and it was developed for humans, and then later on developed as a veterinary vaccine. And extensive studies have been carried out in the U.S. in livestock, cattle, sheep, and goats, and the vaccine was shown to be safe and efficacious. So it's a very good vaccine, and we were fortunate to have the opportunity to collaborate with SUA here in Morogoro, and our plans here are to not only to build infrastructure, which we've already done to a certain extent. We are training graduate students at this point in time. We have renovated a molecular virology laboratory, and we have also constructed a animal holding facility that will allow us to evaluate this vaccine in large animals in this holding facility. We have 3 graduate students on the project. They're all at the Sokoine University of Agriculture here, and they will each be doing a project to support the development of the vaccine. And in Morocco, they will be manufacturing the vaccine, and eventually, hopefully, it's approved for use in Africa. There's a couple of vaccines that have been used in Africa, what we call a formalin kill vaccine. There is an attenuated called Smith-Byrne neurotropic vaccine. Both of these were developed in South Africa, and they have been used for a long time in Africa, but they have problems that are not acceptable by some countries, although they're used in some countries. For example, if you use the Smith-Byrne neurotropic vaccine, it can cause fetal deaths. In other words, animals that are born are dead, or it can cause abortions, especially in sheep. Very high, 90 to 100% of pregnant ewes, gestating ewes, abort. So it's economically, it's very important. And so those are the 2 vaccines. There's a vaccine called Clone 13 that has been licensed in South Africa. Has been used some, but not extensively like the Smith-Byrne vaccine has. So the vaccine we have is a vaccine that was originally produced in vitro in cell culture. It was mutagenized And then we found out that the mutagenesis created an attenuation in the vaccine such that it was no longer virulent, such that it produced disease, and therefore was found to be safe in humans. And then after that, studies have been done in sheep, cattle, and goats in the United States and showed the vaccine to be safe and protective. And then when we were invited to collaborate with SUA, the university here, one of the key questions was to do studies in African animals to demonstrate that the vaccine was also effective in African animals. At that time, the question came up about this vaccine as to whether or not it could be distinguished from a natural infection. In other words, if you vaccinate a sheep and the sheep produces antibody, can you demonstrate that immune response, those antibodies, differ from the antibodies that are found when an animal is infected with the wild-type virus, the virulent virus. And so when we became aware of that, the vaccine was modified such that when you vaccinated an animal, you could distinguish, at least we hope. We're doing a study now to try to determine this, to show that the vaccine, vaccinated animals could be distinguished from those animals that were infected with the wild-type virus, with the virulent virus. And why is this important? Well, in Africa and many countries, when they import animals or want to import animals to the Middle East, for example, they don't want to accept animals that have been naturally infected. So if they've been vaccinated and they're tested and they have antibody, how do you know if they were naturally infected or if they were vaccinated? So we are developing a test that will distinguish, identify the animals that were vaccinated versus those that were naturally infected. We are also just beginning studies here at SUA to evaluate the vaccine to determine if we can vaccinate livestock with a needle-free device, such that these animals will respond as similar to what they would respond to a needle if you used a needle to vaccinate the animal with a vaccine. And we've done studies in rats back at the University of Texas. Methods that we are using, one is an intranasal delivery method, and the other one is through the skin, an intradermal route. And both methods worked in rats, and now we are asking the same question, will they work in sheep, cattle, and goats? And this is going to be the project of 2 graduate students here at SUA. They will evaluate these delivery methods. Well, there's a number of advantages. One is safety. Anytime you're using a needle to vaccinate animals, there's always a risk of sticking yourself with a needle, accidental needle sticks. Another advantage is that when you're vaccinating animals in the field, and especially if they're infected and if you're using the same needle, for example, you can carry over the infection from one animal to another. And so by using a needle-free route of delivery, you can avoid these. Another advantage, and this is, I may stand to be corrected, is that in Africa, the fact that the animal hides are very valuable and they are sold, that sticking a needle in the skin may damage the skin. and therefore reduce the value of the hat. So this is another reason.